Unless it's raining. I said office hour, just meaning, but we don't have to talk biology. We can talk whatever you want. Okay. So, future planning. You got movies. I haven't seen very many movies lately. But older movies I can remember. So, modern music, I don't know anything about. Older music, I know a lot about. Okay. Before we start, any questions? Always open up the questions. Rarely do we get them. Okay, let's start. All right, let's start. We're going to talk about mitosis, which officially is nuclear division, as you know, as opposed to cytokinesis, which is cell division. And as I've said to you before, we often sloppily on purpose because it's easier, we'll use the term mitosis to mean cell division. So, I'll say, oh, the cell goes through a bunch of mitoses or something. That, when I say that, that really is meaning, hey, it's going to do mitosis followed by cell division. But officially, and I just want to make sure you know, officially the word mitosis does not mean cell division, it's nuclear division. And most of the time, it will be followed by cell division. And the official term for cell division is cytokinesis. Okay, but anyway, okay, so we did most of the preliminaries last time. So let's just do a quick review of where we are in terms of a cell that's going to go through cell division. It had to go through the G1 phase of the cell cycle, and then the S phase. What's the S phase?
The synthesis.
Synthesis phase, yes, that's important. So if it's going to go through mitosis, it already had to have gone through G1, S phase, G2, and then it can go into mitosis. So let's just review this. So back in S phase, meanwhile, back in S phase, all the chromosomes are duplicated, right? Duplicated, replicated. Well, I guess in some ways it might be better to say that the chromosomes were they're duplicated. Maybe we should use the word replication for just when you make a new copy of DNA, but it doesn't matter. Duplicated, replicated, it's all the same thing. All the chromosomes were replicated, and so in each chromosome, and let's do this. Let's say for each chromosome there is an exact copy. Or I'll say each chromosome. I'll leave it the way it is. Each chromosome. Now, this is back at X. Each chromosome now has an identical copy. And these two identical copies are held together. Remember that? They're held together. Each chromosome now has an identical copy. And the two identical copies are held together. Okay, let's get started with that. They're held together. And again, remember I said terminology. We get the terminology straight so that when we're using the word, we know what we're talking about. And when they're held together, then those two identical copies of one chromosome, they're not individually called two chromosomes, we call them sister chromatids. In this case, identical sister chromatids. So, and the two identical copies are held together and called identical sister chromatids. I meant to put that in a different color. I just like to use all these colors. So I'm just going to do it like this. Identical sister chromatids. When those identical sister chromatids separate in anaphase, when they separate, then they'll be called chromosomes. Each one will be called a chromosome. So right now, we would say, if I was saying about one of these chromosomes, I say, hey, that's, we're looking at the microscope. I say, hey, that is a chromosome. And you go, but they're two copies of it. Why don't we say those are two chromosomes? That's just not the way we say it. It's all terminology and all that.
They're chromatids while they're attached.
They're chromatids while they're attached. And that's one chromosome made of two sister chromatids. Once these two sister chromatids separate, then they're individual chromosomes. Just terminology. And I said the reasons for it have to do with being able to say how many chromosomes are in a given cell. Hey, and the chromosome number always stays the same, because otherwise they say, well, wait a minute, this has twice as many chromosomes. No, it has the same number of chromosomes, just each identical, each pair of identical chromosomes are held together and they're called chromatids. So we still have the same number of chromosomes. So it's not a big deal in some ways, and it is a big deal in some ways. But it's terminology. So I'll actually put this down. When the chromatids separate. When the chromatids separate, then they will be called individual chromosomes. But of course, they're going to go to opposite poles, and become, they'll end up in separate cells. And if you wanted to really be technical, it's not. But if you wanted to be really technical, you could say, well, back in S phase, they're not even supercoiled, so we wouldn't officially call them a chromosome, because officially a chromosome is what you see in the microscope, and it's all supercoiled. They will get to be that way in prophase, but again, we don't need to be that pedantic about it. We just, that's why sometimes we use the word mitosis just to mean cell division, because it's just easier. And it's a term that's more common. Most people know the word mitosis, but they don't necessarily know the word cytokinesis. It's not that common a term. Anyway, so when the chromatids separate, they will become individual chromosomes, and I'm going to put in parentheses here after this, and will end up in different daughter cells. Okay, so that makes it a little bit, I hope makes it, I want to make this clear, and I know I'm belaboring some of this, saying it many, many times, but I'm hoping by kind of belaboring it, it will sink in what I'm trying to say, and some of the little subtleties I'm trying to get across. Yeah.
Could you maybe draw some circles really quick to show just one becomes two and then.
Well, I am going to, I'll show that with respect to this, but I can certainly do it. I mean, in other words, here's the thing. If you had a cell, okay, so here's a cell, and you had 4 chromosomes. Here's 2 number 1s and 2 number 2s. Okay. Now, you wouldn't actually see them like that in the microscope until we get to prophase, but we're going to do it anyway. So we're in S phase. So in S phase, this is duplicated exactly. This one is. By the way, these are 2 number 1s, but you got one of them from your mom, one from your dad. This is 2 number 2s, one of them from your mom, one from your dad. And these 2 chromosomes would be, those 2 number 1s are called homologous. Remember that term from Monday by chance? They're homologous chromosomes. So we'll say this is the number 1 from mom. So we'll say this is the number 1 from dad. Okay, this is number 2 from mom. And number 2, dad. Okay, now I haven't duplicated. So now we're going to go in S phase, right? So now in S phase, each of these is going to be duplicated. Exactly. So they're going to be like that, it'll be like that. Does that make sense what I'm saying?
Yeah.
It would be like this, like this. Oops, I didn't do a very good job there. Then they're duplicated. So if you really could see it clearly, you go, wait, I see what look like two separate chromosomes here. Well, yeah, but we call them sister chromatids. That way, when we look at it, whatever this cell, whatever cell this is, I would have said originally this cell had 4 chromosomes. Normal chromosome number. There's a term for this, which we haven't given you, so I'll just do it this way. Chromosome. We'll say 4 chromosomes. This cell has 4 chromosomes. And I'll just use the same thing when we started the prophase. This cell has 4 chromosomes. 2 number 1s, 2 number 2s. Okay? Does that make sense if I do it that way?
Yeah, I think so.
And the 2 number. I need to review this anyway, so it's good. This is particularly some of the terms I'm going to use, like the homologous chromosomes. It's not such a big deal today, but not next Monday. Next week is spring break. Oh my God, yes. Next week is spring break. We don't want to forget that.
Viva Las Vegas.
I mean, the people don't go to Fort Lauderdale anymore?
Yeah, that's too far, and Florida is one of the 4 states I've never been to for a reason.
So where do people go to California?
I'm going to Vegas. A lot of people.
I'm going to Bernie Street.
I'm going to Vegas. We were thinking about going to San Diego, but me and my roommate are from there, and we're like, nah. Yeah.
Anyway, don't forget spring break. Don't show up next week. So I was going to say next lecture then, which would be a week and a half from now.
And Vegas
we'll
is cheap as hell.
Meiosis. And meiosis is a special kind of nuclear division. It's going to be followed by cytokinesis also. So it's a special kind of nuclear division, but it's just for the making of what we call germ cells, eggs and sperm. Okay, so it's only for the making of eggs and sperm, and so we have to reduce the total number of chromosomes in half. So we'll worry about that after spring break. But the terminology, like homologous chromosomes, using that term, has become very important to help you remember what's happening with the meiosis. Keep it in mind. So that's why I want to review this terminology. So this cell has 4 chromosomes, and the 2 number 1s, and so the 2 number 1s would be homologous chromosomes. The number 1 from your mom, the number 1 from your dad, they've got the same genetic information. They have the same genes, linear sequence of genes. They've got the same ones. Not identical in the sense that we have variation between people. That's why we have brown eyes and blue eyes, and we're tall and short and different color skin, and you go on and on and on. We have all these different variations. That's fine. But still, the actual genes, like the gene for, I'll say the gene for eye color. It's not that simple because actually it's a gene for an enzyme. But whatever. To make it simple, we say the gene for eye color. They may not have the same gene for eye color, but they still have a gene for eye color in the same place on that whatever the chromosome is. Does that make sense what I'm saying? So again, when I say they're homologous, they're not identical. The number 1 from mom, the number 1 from dad aren't identical, but they've got the same genes in the same sequence. They're just not identical. When the number 1 from your mom is duplicated or replicated, then those 2 sister chromatids are identical. These 2 sister chromatids, they are identical. Okay? But the number 1 from the mom and dad are homologous. So let me just say this, and I'll get your question. So the 2 number 2s are also homologous chromosomes. Yeah.
Does it ever happen just like for some reason where you get one of the chromosomes from like the dad and not from the mom?
Yeah, things happen. We will get to that when we get to genetics. And excuse me, in most cases, they don't survive fetal development. There are a few that do, and we'll talk about that. Yeah, this is particularly true of mammals because the regulation of what's happening in mammals is so careful and exact, and that's the only good explanation we can give. That too much genetic information or not enough presumably because getting too many copies of one enzyme or not enough copies or something, presumably, so that in development, things they don't work out. That's all I can say. But we will get to some, and we'll talk about that. Yeah, we will. It's very interesting. So homologous chromosome idea is important to remember. These are the two number ones from mom, the number one from mom, the number one from dad. Homologous, not equal. Okay.
So if you break down the word to what it is, homo being one, log meaning record, and
Well, the same. And
having
means
qualities is
same,
OUS,
same.
yeah.
Yeah, it's the same. So it's the same sequence of genes. Just there's variations of genes between the mom and dad. All right. So again, back in S phase, and this is back in S phase. All the chromosomes are duplicated. So this would be what it would look like if you could see the chromosomes. I didn't do a very good job of drawing the picture, but anyway, this is what it would look like if you could see the chromosomes back in S phase. Okay. And I put up, I think last time, what is the purpose function? Yeah, these are purpose. Function. What's the function of mitosis? This nuclear division. Well, we're talking about cells dividing. And
distribution of
divides,
genetic material.
we use the word want, which sounds very much like a human emotion, but what the cell wants is when the cell divides is that you have exactly equivalent genetic information in each daughter cell. Remember I said you have a parent cell divides, you have two daughter cells, has nothing to do with gender, it's two different lineages. So what we want with mitosis then, I'll just put this up. I think I had it the last time that I recall. Let's just do it again anyway. So we'll say the function of mitosis. Function of mitosis. To make sure that the two daughter cells, when a cell divides, each daughter cell has the exact copies of all of the DNA of the let's say chromosomes of the chromosomes that the parent cell has. So the function of mitosis is, we'll say, to make sure, that when a cell divides, each daughter cell has the
Genetic.
identical chromosomes that the parent cell has. Parent cell. We're only talking, cells here. We're not talking about, or organisms. We're talking about each cell. So each time a cell divides, we're talking your skin cells are dividing all the time. Your bone marrow cells are dividing. So we're talking any time a cell divides, normal cell division, mitosis followed by normal cell division, of course. So we're really talking mitosis here. But anytime that happens, what we want is that each daughter cell has all the exact same equivalent genetic information, genetic material, chromosomes. Again, if you can use the word genetic material, however you want to say it, I don't care. I just put chromosomes for using that word. Okay, so far.
Yep.
Yeah. If it's not. I'm going to go on.
Now, Colette did a pretty good job of explaining a lot of this in lab, yeah.
And that's the thing, the lab this week is covering this material, so you're getting it lecture and lab, and that's kind of deliberate because then it should sink in better, and you're actually looking at it in the lab, and half of you guys are in the lab already, but you get to look at slides of cells and cell division, and you can see all the stages we're talking about. It's kind of cool. It really is there. It's really fun. In some ways, we make a big deal about this because the big deal was made for so many decades. Because, as I said, when people look at cells, they're just sort of sitting there not doing anything. And suddenly, hey, they're going through mitosis. Something's happening. They go outside. So a lot of time and energy was spent on mitosis. And so that's part of the reason we go through it, but also part of the reason because it's kind of wild how it all works. And there are a lot of little details that are still not understood how it works so well. And for interest's sake, we usually love extremes, if you want to find extremes that are known for various things, like especially about people and stuff like that, the best source is the Guinness Book of World Records. That was started by the McWhorter brothers in Ireland, and I think they might have been in England. I mean, I think they were either Irish or Scottish. I can't remember, but they were actually drinking some Guinness stout at one time at a pub, and one of them started talking about, hey, we need to know who is the best this or the tallest this or the fastest this or whatever. And so they decided they'd actually start collecting all that information. And so that's where this whole Guinness Book of World Records came from. And they did an excellent job, and they would authenticate everything they could. And if it couldn't be authenticated, they would say, okay, this is the way it was reported. We can't authenticate it, so we don't know for sure. So if you want to know, like the tallest person who ever lived, or the shortest person, or whatever, they have all these kinds of things. It doesn't even have to be about people, but of course a lot of it about people. And it's fascinating. I mean, we have this kind of macabre sense of humor and interest about these things, and so some of them are kind of wild. But the point I was going to make, this wasn't from the Guinness Book of World Records, but we tend to like these sorts of extremes. Extremes in terms of chromosome number. I mean, how many chromosomes do humans have?
20.
46, right?
46.
23 pairs? Right? Okay. So what organism has the least number of chromosomes, and what organism has the most? Well, something we haven't talked about, and I wasn't necessarily going to talk about it, but I'll mention it real quickly since I'm on this topic, and that is ants and bees and those kinds of social insects. All the workers, all the workers, they only have half the number of chromosomes. They don't have chromosomes from the mom and dad. They only have chromosomes from the mom, the queen. So they only have half the number of chromosomes. The queen has the normal number, and then a few of the males have the normal number, and then those males can fertilize the queen. But there's something that they do in terms of the development, such that all the workers, which are the vast majority of the bees or ants, whichever one you want to talk about, they have only one set of chromosomes, and they're all derived from the mom. So, at any rate, the point is that there's one particular ant, I don't remember its name, but there's one particular ant that has 2 chromosomes. But that means that the worker ants of that particular species has 1 chromosome. That's all it has, 1 chromosome. We have 46, But there are a lot of reptiles that have in the 70s or in the hundreds. So, I mean, 46 isn't anything magic for us. Chimps and gorillas have 48 chromosomes. In fact, they used to think we had 48. Then later they said
we're short
46,
1 pair. We're
going,
off by 1 pair.
Wait a minute, can't they count? But it turns out that the way they prepared it, one of the set of chromosomes would break, and I thought it was 2. But anyway, the one that has the most is some plant. I don't remember the plant. But there apparently been all these duplications of chromosomes over various, millennia, and this plant has over 600 chromosomes. Huge number of chromosomes. And yet, when the cells divide, they've got to go through the same kind of cell division. I'm going to demonstrate this mitosis with 4 chromosomes so you can see them, Think about that. Okay, that's happening every time one of our cells divide with all 46 chromosomes. But what about that plant with over 600 chromosomes? I mean, it's hard to believe that this thing could work out so exactly for that many chromosomes. But anyway, it does.
That's a lot of codes.
So anyway, okay, now let's start on mitosis. Anyway, Colette and I say anything different, let me know or let her know, and I'll say, okay, one of us lied to you, but not. I don't think we'll say anything different. But we don't get together when we talk about these things. So she's more of a botanist. So she knows a lot about plants. I don't know a whole lot about plants, but I know a lot about molecular biology.
Is molecular biology, is that micro?
Well, it's not actually micro. It's actually dealing with all the reactions and stuff in cells.
Because I just got a book somebody
But-
gave me on
But
cellular.
microbiology deals with microorganisms, but they use a lot of the microorganisms to do a lot of the microbiology, molecular biology studies, because they grow fast and use a lot of viruses because they're little critters that are somewhere between living and not living. Anyway. Okay, so let's do it. We'll do this mitosis with that cell that we just had with the 4 chromosomes. And here's the thing. I mean, if I drew a normal cell, okay, I would say, okay, here's the cell, and here is the nucleus. And most of the time, if you look at a cell, and we did this in lab, I don't know how many labs ago, where you scrape your cheek cell, and we stained it, the cell, and you see the cell, and you see the nucleus didn't look this big, but it was just all dark staining nucleus. You couldn't see anything in it. You wouldn't look at it and go, hey, I see a bunch of chromosomes. No, because the chromosome meaning the chromosomes and all the other material, they're pretty much spread out in the nucleus. You can't really make any detail out. Okay, but if a cell is going to undergo mitosis, what happens, I'm going to erase this because we don't really need this picture, but what's going to happen is, and I mentioned this to you before, but the chromosomes start to coil, and they coil, and they coil, and we call it supercoil. They coil, they're supercoil. So this little tiny thin bit of double helix, which you can't see, even with an electron microscope, you really can't see a double helix. It gets supercoiled, so then you can actually see it in the microscope. And typically, when I just said a cell, you stain the cell and everything, we usually use the nucleus, which contains all the nucleic acid. We usually stain with some kind of a basic, an alkaline stain, a basic stain, because the basic stain is attracted to the acid of the nucleic acid. So that's why you see this nucleus is, generally an interface, for example, which most of the cells are in, just this kind of dark staining thing, but you don't see the structure. But pretty much the first signal that prophase is starting is you start to see individual chromosomes because they start to supercoil, and of course they're picking up that stain, so they become very dark. And you guys have had lab already, you've seen those dark stains. Chromosomes, because that's what's picking up the stain. So let's start with prophase. And we'll say, okay, prophase. It starts with the chromosomes supercoiling. So the chromosomes become visible. Now, what's the state of each chromosome? What are they made of? Each chromosome? 2
Sister chromatids.
Chromatids held together, right? Okay, chromosomes become visible. And remember, because they supercoil. I used to use the example of phone cords. If you had a long phone cord, but nobody has those anymore. Cordless phones at home. But in the old days, we didn't have all those things. We had, this long. The phone cord.
The stretchy cord.
Helix. It's a single helix, not a double helix. It's a single helix. But if you pick it up, put it down, pick it up, it starts getting coiled up, and then it gets all weird coiled, and you can't even pick it up because you have to uncoil it and uncoil it. So, but that's the old days. That shows how old I am. Phone cords. Now, anyway. We even had cameras and you had film in it. And you had to get a negative. And weird. All that's gone.
Yeah, we used to.
Okay, so chromosomes become visible because they supercoil. Now let's. I guess I'll make a little cell. Now, this is going to be the cell, and I was going to put in a nuclear membrane to make it a nucleus. But the next thing that happens is the nuclear membranes, and by the way, these are all happening more or less at the same time. So it's not like, oh, we stopped this, now we start this. It's not step 1, step 2. It's all happening, and this whole process is a continuum. It's starting and continuing until it's done. And so, when you look at it in the microscope, you're looking at a cell where it should stop because the cell's dead, and you look at, hey, what stage is it in right now? And, you know, usually in looking at the cell slides that we use in the microscope, you can find examples of each slide. You sometimes have to look at more than one, let's see, root tip or whitefish, you know, if you look at it, you will see it. But keep in mind it's a continuum. It's not like start step 1, step 2, step 3. We divide it into these steps, but from the cell's perspective, it just starts and goes and it's done. So that you might see anaphase when we get to that when the chromosomes are separating. And you know, they'll, when it just starts, they can call it early anaphase, and then it moves through, and it's almost at the end, you say, oh, it's late anaphase, and then, oh, well, wait, now we're starting telophase, because it reached the, well, anyway, you know, we don't need to worry about that, but what I want you to think of is this is like the entire motion picture of this is happening. We're just stopping it at certain stages and saying, hey, here's what's happening. So don't think that this happens, then this happens, then this. No, it's just that it's happening all at the same time. But we call it prophase. The chromosomes become visible. The nuclear membrane disappears. That's always been kind of a weird thing. You can say, how did it disappear? I thought it was a membrane. Well, yeah, it is. And what happens to it? Well, you know, it'll break up into probably little membranous vesicles and just be hanging out, or maybe just kind of joining some of the other membranous vesicles. I mean, it's membrane. Your cells are constantly making membrane, constantly getting rid of membrane. So it just, I don't know what happens to it. I don't think anyone really knows what happens to it, but it disappears.
Doesn't it just become part of the nucleolus or like absorb into that? Is what it seemed like.
No. It's actually.
Well, it's a phospholipid bilayers.
The proper pronunciation, I want to give you the proper pronunciation. So I'm a little bit of a stickler on pronunciation. Okay. I'm not nearly as big a stickler on spelling it as I should, but it's actually pronounced nucleolus.
Nucleolus?
Yeah, you'll hear it mispronounced nucleolus, but it's properly pronounced nucleolus. The emphasis is on the second syllable, okay?
Okay.
Being funny, but you know, but actually it's nucleolus. But that's actually what the nucleolus is. It's where all the ribosomal RNA is being made. So it's something we weren't necessarily talking about.
Blame the Berkeley
professor. But you'll see
professor.
that, by the way, speaking of the nucleolus, when you look at the onion root tip and you look at the typical onion cells that are in interphase, and the majority of them are in interphase, you can see a light staining nucleus, not really too dark, and then one or more little dark spots, and those one or more little dark spots in the nucleus they are the nucleoli, if there's more than one. And as I said, that's just where there's a lot of RNA is being made, is ribosomal RNA is being made. They're joining ribosomal proteins to make ribosomes. Why are ribosomes important?
What?
Protein synthesis, yes.
Yeah.
We're constantly doing that. But anyway, so I think they just join. I think they're just little vesicles of membrane that they'll be used later. And we will be forming the nuclear membrane back in telophase again. So I don't know if it's the exact same membrane material that makes the new membranes, or it's new membrane. Doesn't really matter. Let me write one more thing here, and then I'll draw the picture. The nuclear membrane disappears, and the centrosomes. I haven't told you about centrosomes. Centrosomes separate and move to opposite poles. Oops. Let me write one more thing.
Take your time.
And because it really continues with this. Move to opposite poles and form a spindle apparatus. Okay. This centrosome. This is a little funny little deal. It's a little.
Is that spindle?
Spindle. Did I not write it right? Spindle. Yeah, spindle apparatus. There's LE right here. Spindle apparatus. I'll draw this in a second, but let me say something about these centrosomes. They're funny little things. They're two little tiny structures. They're kind of at right angles to each other. But back in S phase, well, I don't have S phase up here, but back in S phase, which I said a synthesis phase, and we usually think about it as the synthesis of all the new nucleic acid, replicating all the chromosomes. But it's also synthesis of protein, synthesis of all kinds of other things, including these centrosomes. They're actually duplicated, so you have two sets of these centrosomes. And so these end up, I'm just going to make it real simple. I'm going to put it right here. They end up like this. Okay, so this would be one centrosome, here's the other centrosome. The only point about the centrosome is that they are the center from which these spindle fibers form. The spindle apparatus made of spindle fibers. Now, what are all these spindle fibers? I'll draw them in.
The histone
So
things?
they'll look like this. And there's going to be a lot more. I just am putting several in here, but they're forming from this. Spindle are what they're made of. So those would be that's the spindle apparatus. And these little, I did them in dotted lines. They don't have to be dotted lines. You can make it a straight line, but they're continuous structures. I just did it in dotted lines. So I don't know why I did dotted lines. I did it that way. So I did it that way today. I don't know. Anyway, but we refer to these things as spindle fibers. This whole thing would be the spindle apparatus. These are called spindle fibers. Now, what are these things? Well, they're made of what are called microtubules. Now, this is kind of a wild thing. They're all kinds of little tiny, they're proteins. Okay, they're proteins. Microtubules are proteins. So the spindle fibers are made of protein material. But these little microtubules are proteins that can form or unform depending upon whatever signals they get. And they're actually formed from these little centrosomes. They sometimes call this the nucleating agent. What do they call it? I can't remember. But anyway, they form, and they form out of they said microtubule. I forget what they call it. There's some other term for it, but whatever. But the point is that they form out of these centrosomes, and our chromosomes, they are attached to the spindle fibers. Now there are a lot more spindle fibers than chromosomes. That's okay. But so I'm going to do what we had before. We had two greens and two reds, right? So here's a green one right here. I'll put another green one maybe over here, and here's two reds. Maybe here's a red one right here. Maybe. Red one over here. So one of these green ones would be mom's number one. One of the green ones is dad's number one. Sorry, or I should say, one of them is the number one you got from your mom. One's the number one you got from your dad. Number two you got from your mom. Number two you got from your dad. That's what I'm trying to say there. And why are there two again? Oh God, I've said this so many times, huh? Because each chromosome is made of two
Sister chromatids. Yes.
Okay. And what's the function of mitosis? To make sure that the daughter cells are each going to get a copy
of the genetic information.
One number one from the mom, one number one from the dad, one number two from the mom, one number two from the dad. That's the whole idea. Okay, so look at that. I put it into metaphase already, didn't I? So what I'm going to do then, I'm going to change this a little bit and say this guy maybe is down here, and maybe this one is over here. And in other words, I had them lined up, and I didn't mean to have them lined up. That was my point here. But the point is that the spindle apparatus forms, and they use the term poles, they use the term equator, like the Earth.
Yeah.
That's just for convenience. So let's say the centrosomes separate and move to opposite poles, form the spindle apparatus, and some of the spindle fibers attached to the chromosomes.
So this is after it divides, correct?
This is the beginning. We haven't been divided yet. This is the beginning. Mitosis is nuclear division, but we're not there till we get here. We're just up here.
Okay.
We're just in prophase right now. We're getting ready we're going to go through this, and then we can actually do cell division. So we're getting ready but in order to divide, we need to get, one copy of each of these things up here and one copy down here.
So S phase was before this.
Yes, S phase was back in S phase. Meanwhile, I remember it was G1, S, G2, and then if we're going to,
And then it goes.
But the whole idea of S phase was to duplicate everything, all the chromosomal material, including things like the centrosomes and everything else that you do. talk about it.
Right, and
So let me say one last thing here. This is still all prophase. Okay, we're still in prophase. Centrosomes separate and move to opposite poles and form the spindle apparatus, and some of the spindle fibers. That's what these things are. That's what all these little dotted lines are. Oh, I wrote it. Spindle fibers. Some of the spindle fibers attach to the chromosomes. Thanks. Now, so what did I say that about these? I said the centrosomes. I didn't even write it up there. But the spindle fibers, they said they're called microtubules. Microtubules turn out to be rather important little structures that can form and unform in cells. They help move things around in cells and all that. But we won't need to get into that kind of detail. But just keep in mind, just somewhere way back there, that if you ever hear the word microtubule again, you go, oh God, that old guy was in that bio class said those microtubules were important. But anyway, but they can form and unform. I said that, and that's kind of an important idea because when they attach, you know, they're supposed to attach. I didn't do a terribly good job of attaching, but they said they're going to attach like this. But they're attaching here, and when they attach, then they can, as I said, they can form and unform, and by forming and unforming, they can move these chromosomes. And to begin with, they start moving until they all line up right at the center, right at the equator, if you want to call it that. They call it the equatorial plane, or the metaphasic plate. I usually use the word metaphasic plane, but you could also use the word equatorial plane. But that's the next stage now. So keep in mind that at the moment, these spindle fibers form, they join these chromosomes, they hook on the chromosomes, and they're moving and moving and moving. Like these chromosomes down here, they're going to get pushed up here. This one up here is going to get pushed down here.
getting ready for
Then
moving
we're going to
there.
get into, I guess I'm just going to say, prophase. Now we're in prophase. Metaphase. Metaphase. And that's simple. You know, and when you look at the microscope, it's pretty obvious. It's when all those chromosomes line up with the metaphasic plate, or you could call it the equatorial plane. Line up at the metaphasic plate. I put in parentheses, equatorial plane. Because it's kind of handy to talk about it like the Earth, because you're all familiar with the Earth.
Mm-hmm.
So, in terms of drawing a picture, pretty straightforward. We have this, we have the poles here, we have all these microtubules, spindle fibers. I didn't do dashed lines that time. But all these guys are lined up. So these guys are right here like this. The one over here will be bigger over here. I had green there. Sorry, that's too green. So the reds are, there's the red like this, and I guess that's over. I don't have the exact, you know what I mean.
Yeah.
But they're all lined up right at the equator. What we would think of as the equator.
So you can pull apart the pairs.
Say it again.
So you can pull apart. That's where it pulls.
Exactly. Get them all lined up, then we can separate them, which is anaphase. But before we do that, this term is metaphasic. This must be I don't know if you can see that. I see metaphasic plate. It's just a term for this. You know, it's an imaginary plate, just the way the equator is an imaginary circle going right around the middle of the Earth. So that's what that metaphasic plate would be, an imaginary plate right at the equator, and that's why you call it equatorial. Okay, that's straightforward. Nothing tricky about that. Anaphase is when they're being pulled apart. Okay, so let's just I just do it right below it. So anaphase and anaphase. These words are kind of funny. I mean, pro means before, so it's the beginning. Meta usually means beyond, but it doesn't always mean beyond. It can mean other things too. So Anaphase means back, so it's like, hey, you're going back towards the poles. Telophase usually means the end. So, telos. You can use, if you ever use the word teleology, that is referring to purpose, some purpose. If you give a teleological explanation about something, you're given some purpose for whatever's happening. So, in other words, the end process, you're saying there's some purpose involved. So, it really means the word end comes from the word meaning end. Anyway, anaphase is easy enough to recognize this because when these guys are being pulled apart, one of each of the sister chromatids is pulled towards one pole, the other is pulled towards the other pole. Okay, so anaphase, the sister chromatids are pulled apart. And start moving towards opposite poles. How are they moving? It's those microtubules. As I said, they're in some cases that if you're forming in this case, I think the microtubule is attached to it is unforming, and so the whole microtubule is getting shorter, pulling one of them that way, and this one's getting shorter, pulling it the other way. So in some cases, microtubules can grow and push something. In other cases, they can be attached to something, they can shrink and pull it away. Shrink meaning they're ungrowing, they're getting rid of stuff. Anyway, the sister chromatids are pulled apart and start moving towards opposite poles. But now that they pulled apart, so those two identical sister chromatids are pulled apart. What do we now call them? Chromosomes. Now we can call them the old term chromosome. Chromatids are called, it's just terminology, chromosomes. So drawing the picture, I have to make this picture a little bit smaller. This simple picture, I'll just put green on it. I'll do it about halfway. There's one here, there's one down here. Here, this is here, this is here. What should we like to say distance here? And the red ones here, and over here, here, and over here, something like that. And you know, it's kind of neat because they're being pulled through the cytoplasm. That's why they're actually facing. I mean, notice I had them both facing the same way here, but here they're actually facing obsoletes because when they're actually being pulled through the cytoplasm, it's like the arms are they're actually pushing against the cytoplasm, so they actually are flapping one way versus the other. Now, when you look in the microscope, I didn't do a very good job. These are supposed to be sort of semi-lined up here. When you look in the microscope at these, and you know, you're supposed to find examples of anaphase, examples of telophase, examples of metaphase, all that. And sometimes, you know, once they've separated, you say, oh, that's definitely anaphase. What about if they're up here? Is it still anaphase? Probably. We officially say it's telophase when they reach the pole. But sometimes they'll be really close, and you go, well, is it telophase or is it anaphase? And we'll go, yeah, you could say it's late anaphase or early telophase. I mean, you know, in other words, it's not a big deal as long as you understand the process. Yeah.
In metaphase, then, are they chromatids or chromosomes?
So in metaphase, there's still that's one chromosome right there, made of two sister chromatids. Down here, hey, those are two chromosomes.
Okay.
Terminology. So
Okay.
they haven't changed anything, you know. It's just the way they describe it. And again, it has to do. You want me to say it again? Yeah. Okay. So here, right here, that's one chromosome. Okay, that's one chromosome. That's one. So there's right now there's 4 chromosomes. But each chromosome is made of 2 sister chromatids. Same thing here. Same thing with metaphase. 4 chromosomes, each one made of 2 sister chromatids. Down here, now that they've separated, we say, oh, there's 4 chromosomes up here, 4 chromosomes down here. And then the cell's going to divide, so we'll have separate one set down here.
They're in pairs until annotated.
Yeah, they're in, what'd you say?
They're in pairs until annotated.
Intel anaphase, yes. They're in this pairs of two sister chromatids, yeah. Okay, so now telophase, they reach the poles. But that's not all that happens. This is like that, so you know that this is going with whom. Telophase, the chromosomes reach the pole. Now I'm calling them the chromosomes because that's their proper term. The chromosomes reach the poles. But then all the things that happen in prophase, gets reversed. So what are some things that happen in prophase? Well, we said the chromosomes become visible. Well, they start to uncoil. So the chromosomes reach the poles. Again, it's not necessarily step. It's all hand happening at the same time. Chromosomes reach the poles and start uncoiling. It doesn't just happen immediately, of course. And then what's the next thing we have? The nuclear membrane disappears. The nuclear membrane is going to reappear. But in this case, we're not there yet. I'll draw the picture, but we're going to have two nuclear membranes. Let me draw it. That's just wrong. That's too big. Okay, so we had all this stuff up here, but now we pretty much have the chromosomes. So we have what? Two green ones, two red ones. They're all the poles. Now I'm not having them uncoiling in my picture. I want you to see them. Okay, but now the nuclear membranes start to reappear. Again, it doesn't just happen immediately, but they will, and we'll get nuclear membrane reappearing in a little while. So that's supposed to be nuclear membrane. That's just put it. So nuclear membranes reappear. I should say reform. And again, this doesn't all happen exactly at the same time. This is happening over a period of however long, maybe 15 minutes or something like that. Nuclear membranes reform. That whole spindle apparatus has started to, disappear. I mean, it's started to break down, or however you say it. So the spindle apparatus. I'm going to say, make it easier. Apparatus. The spindle apparatus. I'll just say disappears. How about that? But notice, just for interest's sake, it's not something that's ever going to appear to test for my purposes. There's a centrosome up here and a centrosome down here. And the cell's going to divide in the center. So each cell is going to get a complete copy of the chromosomal material. It's going to get a complete copy of, it's going to have a separate spindle, or I should say centrosome. So that then the next time it divides, that centrosome can, it'll divide, in the S phase it'll double, and then you'll separate, blah, 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 blah. You don't need to worry about that. And that's all that kind of stuff happens. And then cytokinesis occurs. Then cytokinesis. Now, it's a little different from animal cells and plant cells because animal cells have a cell membrane. Plants have a cell membrane surrounded by cell wall. So it's a little trickier with the plant cell. So we'll talk about the plant cell in a minute. But what happens in cytokinesis, and then, again, I'm saying, you have this, then you have this. But actually, cytokinesis will start before everything is finished. What I put up there for what's happening with telophase, it may not be finished yet when cytokinesis happens. That's fine. I mean, don't think it has to stop this and then go to this. It's not that simple. But what happens right at the equator, there's a thing called, I'll have this, I'll put it down here, but I'm going to put it up here. There's what they call a contractile ring forms. contractile ring. This is, well, that's what I'm saying. This is really officially, you can say, oh, this is what happened to cytokinesis. But before you finish all this, you can start. You can start doing the cytokinesis before everything is completely finished. In other words, you still, these chromosomes will not have totally uncoiled yet. You might still see a little bit of the spindle apparatus. The nuclear membranes probably will have formed, but maybe not totally. It's just not an absolute here. So with the cytokinesis, that contractile ring forms. What the heck is a contractile ring? What kind of material do you think it might be made of if it can contract? And by contracting, it's shortening. What'd you say? I said muscles. I said protein. Yeah, protein. Good. Yeah, protein. And the analogy I would give you take your belt that's right in the center here, just start tightening it and tightening it and tightening. You start squeezing. That's really what's happening with the contractile ring. Is it's these proteins that are shortening. And yeah, I don't remember for sure. I don't know if I've ever actually looked it up, but there's a reasonable chance it might be made of some similar proteins to muscle proteins. But I don't want to say that's the case because I can't really remember offhand. But whatever, it does shorten. It gets small, gets smaller and smaller, and it's a ring right around the center. And it just starts squeezing and squeezing and squeezing. So it would start looking like, let's say it's up here. It's starting here because I put the contractile ring in, but then it's going to be okay, it's going to look like that, it's just pitching it off. I mean, it's getting smaller and smaller, squeezing. And then eventually, of course, it's going to pinch off totally. And I know in the lab on Monday I saw a couple of cells that you could tell they had just pinched off and they were just beginning to separate when they have to make the slide. So it's kind of cool because you could see that they were just pulling away from each other. But anyway, they contractile ring forms, pinching off, or pinching a little, we could say the original cell, the original parent cell into two daughter cells. And these are cell membranes. Cell membranes, phospholipids and proteins. They can, you can put two membranes together to form a big one. You can separate them and they'll pull together. The neat thing about those phospholipids is they like each other. So you can pinch them off, and they'll, the membranes will just close up on themselves, and they'll be separate cells. Contractile ring forms and pinches the original cell. Into two cells. I mean, that's as detailed as we need to worry about mitosis, or about cytokinesis. In an animal cell, pretty straightforward. Plant cells are a little tricky.
I mean, me. I had the best game.
It's not as bad as it seems now, but in the old days, before these buildings, that means a year ago, and before, it's sort of, I'm sorry, I'm not trying to make any nasty remarks about groups of people, but you could always tell the AR kids, the Academy of Records, because they're high school kids, and they would have classes in the physical science building, and when they get out, I'd be maybe in a lecture or maybe in a lab, and you could always tell when the high school kids got out of class, or maybe going over to the cafeteria or something, because they were so loud and noisy. Typical kids. I mean, it's great.
Yeah.
But you always tell there's so much noise in college kids in there. And they usually hang out in bigger groups too, anyway. So a lot louder. But anyway, okay. So does that? Before I talk about cytokinesis in plant cells, which I can sort of explain, but there's still some kind of, it's not absolutely clear, but you'll see what happens in a second. But are there questions about any of this? I want to make sure it's clear to you.
It's just that this is all only eukaryotic cells, right?
Only eukaryotic cells. Because what's the deal about what kinds of organisms are prokaryotic again?
Bacteria.
Blue-green bacteria. Okay, and what do they have? They don't have pairs of chromosomes. They have one circular chromosome.
Mhm.
Small too. Because they're smaller organisms, they don't have nearly as many genes. But one circular chromosome. So they do, that when they divide, they will divide in, so there'll be two circles, and then they don't do mitosis, they don't have spindle apparatus, they don't do all those phases, that they'll go into two, and then the cell will just divide, and then they'll be separate. And it's called binary fission, term for it. I won't give this to you on the board. It's called binary fission, binary meaning just splitting it into two. Fission meaning splitting. You've heard fission was what happened. That's the atomic bomb. They talk about fission because the uranium atoms or plutonium atoms are splitting. Anyway, as opposed to fusion, where they're trying to get
Joining,
joined together, fusing as opposed to fission, splitting. Okay, but anyway, whatever. Okay, so any questions on this thing? Because I want to make sure it's clear. Eukaryotic cells, everyone. And this is what I've shown you here. The way I did this would be some kind of an animal cell. Or it could be a protozoan or something like that, but something that just has cell membrane, doesn't have a cell wall. If we're talking about plant cell, it'll still do the same basic things. Everything will be the same, except for the cytokinesis, because they have a cell membrane and a cell wall. All cells have cell membranes, right? But they have a cell membrane and a cell wall. And what's a cell wall made of?
Phospholipid.
Cellulose? No, cell wall.
Oh.
Cellulose?
Yeah.
So you've got to have that cell wall. So let me show you. Gee, I'd like to lead us up to the next class. This is nice. Save a lot of writing. But I'll tell you what. Let's just erase this part and do the cytokinesis idea for plant cell. So we'll say in a plant cell. So we'll take, here's the original cell. Well, I'll make it a little longer. Here's the original cell, and we're going to want it to look like, well, let me draw this first. So this is, in black I'm doing cell wall. Let's do, if you can see the red, let's do the red with the cell membrane, okay? So this is what we want it to look like when cytokinesis occurs. So in red is cell membrane. Oops.
Are plant cells always kind of rectangular or square like that?
Much more so, yes. Much more so than say animal cells.
Yeah.
Because animal cells are more flexible.
Yeah.
So yeah, pretty much. Okay, and then so in red I'm trying to do cell membrane on the inside, cell wall on the outside. So this is what you want to end up with is two cells, but you want the cell wall in between the two cells with the cell membrane and completed. Well, it's a little tricky because you can't just pinch it off. It's solid because of the cellulose wall. You don't just pinch it off. So what happens in plant cells is that in the center, right in that equatorial plane, if you want to call it that, little vesicles form. I'm going to do it like this. Well, no, I shouldn't do it that way. I should do it this way. You get little vesicles forming. Oops, they're not there. And in that vesicle will be some cellulose material forming too. I don't know if you probably can't see that in the back, can you? I'm trying to put. I'll make it bigger. Make it much bigger. I can take away this arrow too. Okay, so I'm going to make a couple of big vesicles just so you can see them. Okay, two big vesicles. It's actually a whole bunch of little tiny ones, but I'm deliberately doing two big ones. And you get some little cellulose material. So these would be vesicles. I should say membranous vesicles. Let's write this again. Membranous vesicles. Containing cellulose. form in the center of the cell. Now, of course, we're looking at it two-dimensionally, and it's three-dimensional, so I mean, so you have vesicles all along a plane here, all along the plane. So that's what these little guys I'm trying to show you, those little vesicles, and red is supposed to be membranes, the membranous vesicle, and inside the black is supposed to be the some cellulose is forming. And they're really tiny, not big like this. There are a whole bunch of them, and there's a plane of them because the cell is fairly wide here. But they're just forming, and they're forming more of them, and they're just moving towards the edges. And as they move towards the edges, well, what about that red stuff? Membrane. And as it moves towards the edges, it can just join with the membrane that's here. And the membranes all along, and these vesicles can join. You know what I mean? This membrane material can join. And then there's this cellulose material that's in between, and it can just continue to grow and eventually fill in between. Now, so what am I saying here? So if I took a couple of these, I'll take more of them. So we have, here's one vesicle, here's another vesicle, here's another vesicle like that. And it's got, you know, some cellulose material forming inside like that. And then, so here was, we're getting close to this, right? So here's the membrane like this. I'm not showing the whole cell, I'm just showing you this little bit right in here. Okay? See what I'm saying?
Yeah.
As this grows a little further, this membrane can join up over, the membrane of this vesicle can join up here. These membranes are going to kind of join up. These membranes are going to kind of join up because they're actually growing into each other. They're all joining up. So eventually what's going to happen then, you're going to have membrane forming here like this, membrane forming here like this. Because you see where this forming here is going to kind of
Pinch off, yeah.
Because all this cellulose material is going to be in between, and it's going to kind of fill in like that. That's kind of the idea. And of course, it'll grow all the way to hit. To hit this. You don't really need to worry about the detail on it. I just want to kind of show you sort of how it works. It's not a real big deal. It's just it's a little different because
The
the cell wall. And the main thing to think about is just that there are these membranous vesicles that form right in that equatorial plane, and they're containing cellulose on the inside and this membrane material on the outside. The membrane, as they fuse, they can just form two sets of membranes, the upper part and the lower part, they just form that cellulose in between. Sort of makes sense?
Mm-hmm.
It's about the best I can do for explaining it.
Seems like cells are
Any
smart.
question?
I was trying to think of how to pull apart. I was wondering if it grew like a.
Wait, who pulled the ball apart?
Well, I mean, how to make them two separate cells, or do they just stay fused?
No, they
together?
stay right there. Yeah, they're still right there. Yeah. They're not pulling apart the way the animal cell might be. And you know, you've got this solid wall here.
So, I mean, since they're going to be growing together anyway, I'm just saying that.
Yeah. And of course, you know, like that root tip, if you were to lap, you know, they're going to be growing like this. This cell will get bigger and bigger, and they're pushing that root tip down. And you know, as these guys get longer, they're pushing all the ones above them further down. You know, and each of these is growing every so often. Yeah.
So they share that cell wall that's inside of it.
Yes, that's right. They share the cell wall, right? And you know, there are all kinds of little things that you know we don't need to worry about for our details. But you know, if you take botany or something, there are little tiny holes in that cell wall that allow various things, fluid, nutrients or whatever to go from one cell to another. So there are little. What are those called? Plasmodesmata or something like that? I don't know. I think that's the term, if I remember correctly. Dredging that up from the last botany course I had, you wouldn't, before most of your parents were born. But I think it's plasmodesmata. I think it's a term for little openings here that allow things to go through. That could mean something else too. I can't remember. Questions? Any questions on this?
No.
We did pretty well, actually. What I want to do, and there's a little bit of time left, because I wasn't sure exactly how long it was going to take. Let me just give you a preview. I won't even write it down, but a preview of what we're going to talk about when you come back from Fort Lauderdale or wherever you're going, Las Vegas or
Las
wherever you're
Vegas,
going.
yeah.
If you can still remember anything that happened this week, and I'm hoping you.
That's why there's the video.
That's right. That's right.
I don't have to remember.
Materials, right?
Yeah.
But anyway, here's the idea. We talk about mitosis inside of Mimesis. I'm going to use the word mitosis in the sloppy way. Just think of it as cell division, even though officially it's not, because it's easier to talk that way. So we were talking about just normal cell division. When a cell's going to divide, skin cell's going to divide, or amoeba's going to divide, or whatever. Normal cell division. And what was the idea of mitosis? To make sure that, I erased it. To make sure that when a cell divides, each daughter cell gets the normal component of all the nuclear materials. Every one of the chromosomes. Alright, that's called mitosis. But we want to talk next time about how you form eggs and sperm, so-called germ cells. By the way, the word germ, I know we use the word germ to talk about microorganisms that cause disease, but the word germ actually comes from the word meaning to form something new. So when we use the word germ cell in biology, not talking microbes or something where they cause disease, we're talking about cells that are going to form the new offspring. So the germ cells, when we say that, we mean forming eggs and sperm. Okay.
So like wheat germ.
Like the wheat germ is what's going to is used to form the plant. Yeah. So anyway, here's the deal. Glad you guys are putting all your books away. You never know. It might be something fascinating you need to write down. Okay, put your books away. Just teasing. Anyway, the point is that when we form eggs and sperm, think about this for a minute. How many chromosomes do humans have?
46, 23
right?
pair.
23 pairs, 46 chromosomes. If eggs and sperm both had 46 chromosomes and they joined, then suddenly, my God, you'd have a cell with 92 chromosomes. And then if they formed eggs and sperm, then you have a cell with 184 chromosomes. Whoa! So it became obvious, to early biologists that can't happen. They must do something else. In other words, there must be a way when you form eggs and sperm where you only have half the number of chromosomes. And that's exactly what we'll be talking about. And that's the process of meiosis. Okay. And I'll put the word down just so you see it, because it's spelled differently from meiosis, but you have to be careful when you say it. So you have to be careful when you see it, so that you or hear it. If you're hearing either mitosis, which we think of as normal cell division, or meiosis, that's a special kind of division. It's called a reduction division, and it's for the formation of eggs and sperm. And the whole idea when they use the word reduction is you want to get the total number of chromosomes in half, but in a nice way. So, for example, we have 46 chromosomes, 23 pairs. Of each of those pairs, one's from your mom, one's from your dad. Well, when we form eggs and sperm, we want 23 chromosomes. One number 1, one number 2, one number 3, one number 4. It turns out, though number 1 won't be exactly the number 1 from your mom, it won't be exactly the number 1 from your dad. It'll be some mixture, and we'll talk about that. It's kind of interesting how that works out. But there are ways to make sure there's more variation. But officially, that's the whole idea of forming eggs and sperm in the so-called meiosis, cutting the number of chromosomes in half. So that then when you have an egg with 23 chromosomes, a sperm with 23 chromosomes, they unite, you get a new cell with 46 chromosomes back to the normal kind. And we'll introduce some other words, haploid and diploid and all that. Question?
Wouldn't you have a question about, for instance, eye color? When you get that from your parents?
Right.
What if, in my case, I got my eye color from, my great uncle or something? So
we'll have to talk about this later. No, I'm just kidding. I'm just kidding. I'm kidding you.
Is that due to, how the enzymes are on the gene?
Yeah, no, and actually you didn't get it from your great uncle, maybe the same as your great uncle. You got it from your parents, but it's how the genes were expressed. And it may be expressed like your great uncle. But yes, you're right. It has to do with what you've inherited from each parent, and then how they blend when they are, being expressed. And it's kind of neat the way it works. I mean, it's kind of fun. And we will get into that, not next week, but when we get into Mendelian genetics, we'll get into that. And we'll talk about various things.
Dominant and recessive genes and stuff like that.
But anyway, yeah, we'll do that. All right, so have a good spring break, you guys. See you in a week.
All right. All right, guys. See you after the break. Turn off.
Genetic copies.
Hello.